Yes. Okay. Yeah, Neha, now I can see you. Hello, Neha.
Hello.
Yeah, I can see you. Okay, it's okay. It's
Okay.
fine.
You can hear me?
Yes, yes.
Okay. Good morning, doctor.
Good morning. Good morning.
Muthamilarasan. The name is
Sorry to.
South Indian, so
Yeah, sir, I would like you to at least pronounce it for me once so that I'm not doing it wrong. While I.
Muthamilarasan.
Sorry?
Muthamilarasan. It's not possible to pronounce. So yeah, I keep it short. Muthu.
So I can call you by
Muthamilarasan.
that if it
Yeah,
is
yeah,
fine with you.
Muthu. Yeah,
Okay.
that is perfectly fine.
Fine. That would be
Thank you,
really
thank
nice
you.
of you. Yeah.
Okay,
So
I'm
I
just
think.
allowed to attendees to.
Yeah. Sure,
uh,
sure.
Okay, because we are in 11.
Yes.
Okay, they are coming. We are also in the YouTube live.
So we'll be taking up the question and answers.
After no, just finish of the stock at last day, what we do now, we select few question in question answer round. Those are the real question, and hand it over to the sir, and we going few of them, and then we start our interview session. So we just finish the whole talk, then we start the interview. Today we are officially getting 1,697 registration. Actually, is the
That's cool.
registration process. But obviously, viewers will be lesser than that. But okay, man, it's a huge number of expectations. You can say.
Yes.
So
It's
I
necessary
send.
for us to promote plant science this way. We never knew there are so many patrons available. And we started this.
Yes. Okay, it's 11 first, so we start. We can start and let them just.
Yeah, they'll keep on
come in.
joining.
Okay, so I just starting the recording. Okay.
Yeah.
Good morning. I am Shubhabrata Majumdar. Today we're welcome all of you in Zoom and YouTube platform. It's another webinar. Is a live webinar from our end, and we expect your patience because we are giving to the password during the question answer round. So please don't panic or excited. It or definitely it will be given to you during the question answer round. And today we have Neha introduce our speaker, and Shoma will going to the interview session. So I hand it over to Neha to introduce our speaker. To Neha.
Yeah. Good morning, everybody. Thank you for joining us here on the Bioingene webinar talk today by Dr. Muthamilarasan, who has earned his postgraduate degree in biotechnology from Madurai Kamaraj University and got Ph.D. in plant biotechnology from NIPGR New Delhi. His current research focuses on structural and functional genomic study towards the improvement of foxtail millet. On the structural genomics aspect, his group is developing different large-scale molecular markers for use in genomics-assisted breeding. From the functional genomics perspective, His research group is characterizing important genes and gene families to delineate their precise roles in conferring climate resilient traits. In his area of research, Dr. Muthu has published 30 original research, 12 reviews, 11 book chapters, and 9 popular articles. The citation index of his publications is 1904, H index of 24, and his item index is 32. In recognition of his scientific contributions, he has been awarded several national and international awards, such as INSA Young Scientist Medal in 2018, NASI Young Scientist Platinum Jubilee Award in 2018, USIF Fulbright Alumni Award in 2019, Indo-Australian Career Boosting Gold Fellowship in 2017, Fulbright Nehru Doctoral Research Fellowship in 2015, etc. Recently, he has been selected as a member of the prestigious Indian National Young Academy of Sciences, India. In addition to doing science, Dr. Muthu routinely travels to the rural areas of the nation to deliver popular lectures in the areas of career development to school and college students. His books on fellowship and funding opportunities for school students is popular among the educationalists and policymakers. With this, I request Dr. Muthu to share his screen for starting the talk. Over to you,
Yeah.
sir.
Thank you, ma'am. Thank you. Yeah, is that visible?
Yes.
Okay. So, good morning, everyone. Uh, it's nice meeting you all through this virtual platform. And thanks to Team Bioingene for providing me this opportunity. And you people are really doing a great job. I could see the updates on Facebook, emails, and everything. And I never imagined that I will be there as invited resource person to deliver a talk, because I've seen several big shots where talking there, and even my PhD supervisor Dr. Manoj Prasad is yet to deliver a talk in your platform. So I have to leave some space for him also. I should not cover the complete foxtail millet story in my slides. So we came to an agreement that we will split it into part A, part B. So I will talk, or I will make the base on which he will go in detail to tell how far we have worked on this crop, which was once orphan, or which was once underutilized, which was once neglected in terms of research. But we picked that crop and we worked. We made tremendous progress in this crop in terms of developing genetic and genomic resources. So yeah, so today my talk is on this development of genetic and genomic resources in foxtail millet, where I'm going to tell you the story of how we began to work on this crop and what our work has contributed to this crop, and how this can be used as a model to develop similar resources in other minor millets for their genetic improvement or downstream studies. I don't have much to show, to be frank, because the work was started only in 2012, and it is now being carried forward by my juniors at Dr. Manoj Prasad's lab and my own students at University of Hyderabad. But still, we will give you, or I will try to give you an idea how to mainstream an underutilized crop bringing it to the limelight of research. So as we all know, millets is a common term to categorize small seeded grasses that are often called as dryland cereals. The grasses that are referred to as millets are categorized into two major groups. One is major millets, other one is minor millets. Sometimes sorghum is also categorized in the major millets, but I have not included it because predominantly or typically we Indians include sorghum under major millets. And in minor millets, there could be proso, kodo, barnyard, little, and other pseudo millets. Yeah. So in 2018, our central government has recognized these millets as nutri-cereals. This is a screenshot of this gazette. And our Indian, we requested the United Nations to announce the year 2023 as International Year of Millets, which has also been formally accepted. So to know more about the small millets, I would recommend the viewers to read this article published 2 days ago, which is a comprehensive review on small millets covering almost all the aspects right from genetic genomic resources, breedings, transgene-based research, etc. So you might be knowing that India is the second largest producer of millets next to China. And in terms of area, Rajasthan and Maharashtra tops in millet cultivation. However, in terms of yield, Uttarakhand and Tamil Nadu leads the race. So, but still we are the second largest producer while the topmost is China. And among millets, the crop in which we are working is this foxtail millet. Foxtail millet or Setaria italica is a diploid crop having approximately 500 MB genome in 9 chromosomes. Its life cycle is very similar to other cereals like rice. And of course, by belonging to the grass family, it shows or it shares similarity or greater degree of genetic similarity with cereals like sorghum, maize, rice, and bioenergy grasses like switchgrass, napier grass, etc. Being a C4 crop, foxtail millet also possesses high degree of adaptation to diverse or adverse environments. So I have split my today's talk into two sections, one as the progress made in structural genomics and the other as the progress made in functional genomics. So we all know that genetic and genomic resources are imperative for any crop improvement program, where genetic resources serve as the primary input for breeding, and these genomic resources facilitate the efficient characterization of the genetic resources and their utilization in crop improvement. In foxtail millet, excellent genetic resources is available in NBPGR, in ICRISAT. Even we ourselves have around 500 germplasms of foxtail millet global collection. So genetic resources is not an issue in foxtail millet, but when we started working on using this genetic resources, one difficulty we faced that was the lack of molecular markers and physical or genetic maps. When I joined for PhD in the year 2012, there were only 2 papers published in foxtail millet. One was reporting 26 ESSRs and the other one was reporting 98 ILP markers. So what we thought is like, first thing we have to do is what we thought, like, we will first develop large scale genome molecular markers and then we will go for association mapping or genome-wide association studies. So in that direction, we started developing large scale molecular markers where we used the publicly available transcript data, because that time we didn't have much resources to sequence our own transcriptomes. So we had to rely on the publicly available transcriptome data, where we used them, segregated into 2 coding sequences and intronic sequences. And in the coding sequences, we run this microsatellite identification tool to identify microsatellite markers. And in the intronic sequences, we used rice as a reference to develop markers that flanks the polymorphic introns. So we developed more than 500 SSRs and 5,000 ILP markers, which we further used for genotyping assays. On the other hand, we also realized that the repeat junctions of transposable elements are unique, and these could be genome specific, and they can therefore be treated as single copy markers in the genome. So what we did is, like, first thing, we identified the transposable elements present in foxtail millet genome using different pipelines like LTR finder and repeat masker, and then we exploited the repeat junctions to develop primers or to design primers which served as transposable element based markers. So by this way, we developed up to 20,000 repeat junction based markers, which we again used for genotypic assays. So by this way, we now developed several markers, 20,000 of TE based, 176 of microRNA based, 5,000 plus ILP based, and 400 plus ESSRs. So these markers certainly showed high levels of transferability, which highlighted the application of these markers in comparative genome mapping and evolutionary studies in other grass species, particularly those lack genome sequence information or genetic maps. And the developed markers were also used to construct or analyze genetic relationships and genetic diversity of millets and non-millet species. the results showed that minimal number of markers were able to discriminate between millets and non-millet species, as you could see here. Maximum synteny of these markers were observed between foxtail millet and sorghum, followed by foxtail millet maize and foxtail millet rice. And the map clearly indicated the decline in synteny with increase in phylogenetic distance between these crops. And even in certain cases, you could see the nested chromosomal fusion, which was frequently observed in the grass genomes. So yet further, as now we have developed several markers, now we have to use these markers in identifying a gene or in developing a valid marker which is straight away linked to some trait. So for this we went and we collected 184 foxtail millet accessions from diverse geographical locations around the globe. You could see from where and all we have got the germplasm. And what we did from 2013 onwards, we did phenotyping in our NAPGR fields for 10 yield contributing agronomic traits for 3 consecutive years. So now we have the phenotypic data, on the other hand, we have the genotypic data. Then we went for association mapping, where we identified 8 markers associated with 9 different agronomic traits, contributing to this much percentage of phenotypic variation. That was perfectly fine. By this time, what happened is NGS came into the picture, and when NGS came into the picture, we were able to go for whole genome resequencing or low throughput sequencing. So then what we did, although we have published whatever we have done so far in different journals, then we went for DD-RAD sequencing, double digest restriction assisted sequencing of DNA. So by this way, DD-RAD coupled with Illumina platform fetched us around 30,000 high quality SNPs, which we used for mapping which we used for performing GWAS with our phenotypic data. So one thing, since we already had the phenotypic data, we did GWAS, we identified 81 marker trait associations involving 79 SNPs that were identified, and we published that in scientific reports. If you could see, several genes were still unknown, and students are working on these unknown genes to know what exactly these genes have role in regulating grain yield or 1000 grain weight or flag leaf width, etc., etc. Even in one case, this ABC2 type transporter, this has been recently characterized for its role in grain size in chickpea recently. We have also got a kind of its ortholog, so we are also trying to work on this aspect. On the other hand, you might be knowing that foxtail millet or millets in general are rich in micro and macronutrients compared to other millets and major cereals. So what we did is we used ICP-MS to analyze 10 micronutrient content in different millet grains. As you could see this data, finger millet is obviously rich in calcium, foxtail millet is rich in phosphorus, magnesium, and sulfur, even zinc. So yeah, millets are obviously rich in micro and macronutrients. So what we did, we estimated these 10 micronutrient elements in our 184 plus. After that, we even got a little more. So now, by that time, we had around 200 foxtail millet germplasm. So we analyzed the micronutrient content in those 200 germplasm, and then we performed GWAS, where we identified 74 marker trait associations for the 10 elements. Yeah, it's even here you could see that different markers which were associated with different micronutrient elements and the corresponding genes where these SNPs are present. So the students are still working on characterizing each of these genes and the respective markers to identify their relevance in regulating the content of these micronutrients in foxtail millet grain. And this story was published in the Journal of Cereal Science. So this is where we are going on. When we started, there was no marker. Now no markers are maybe less than 100 or 120 markers. So first thing we had to do is to develop markers. Yes, we developed ESSRs, we developed ILPs, we developed miRNA-based markers, we even developed transposon element-based markers. Then we tried to use these markers to study the marker data association. And then GWAS came into picture. We were able to do DDRAD sec for genotyping by sequencing. And then by this way, we have identified the SNP is linked to several traits, including micronutrients, yield contributing agronomic traits, etc., etc. Now we are further characterizing these genes. So since that work is still in progress, I'm not showing you what we are doing or what is actually going on in the lab. But up to here, you should be, clear that when we started, there was no resource at all. But in a span of 3 to 4 years, we were able to develop several markers, and we were also able to demonstrate the utility of these markers in genomics-assisted breeding. So then we didn't stop by publishing by ourselves. What we did, we constructed a marker database and we put all the marker resources in that database, and we kept it open for the global research community to access. Although now this database is not functional due to some server issue, but we are now trying to restart it in a different server. So very soon it will be online. So in this marker database, one can access the details of each and every marker we have developed. Detail in the sense, it could give you even the chromosomal number, physical position, what is the motif, how many times it has been repeated, is there any other information linked to that, and you could even visualize the physical location of each of the markers in an interactive physical map, which was created using a portal or a tool called CMap. We have even constructed a comparative genome mapping tool there. You could see the arrangement of markers on foxtail millet chromosome with maize chromosome, which shows this nested chromosomal fusion, which was frequently observed in grass genomes during the course of evolution. So such comprehensive database was also being developed by us, which we kept it open for the global research community to access and use these markers. And yes, ICR, IAMR, TNAU Coimbatore, there is one institute, university in Andhra Pradesh, Yogi Vemana University. They are all using our markers to characterize their germplasm. So the resources which we developed are, of course, being used by the global research community. That is the best part of this. So with this, I'm just moving to the functional genomics aspect, where it is a long way to go, but still I have a little bit of data which I can show you, like how we established, or we were the first people to do functional genomic studies in foxtail millet. Before the genome sequence was released, two of my seniors, Dr. Charulata and Dr. Swati Puranik, they were working on, they worked on two different genes. One is DREB2 and the other one was NAC. In DREB2, they identified an SNP which was linked to the stress tolerance trait, and that was published in JXP before my entry into the lab. And this is about the NAC protein, SINAC. The other senior, Dr. Swati Puranik, she characterized this NAC transcription factors in foxtail millet. When I joined Dr. Manoj Prasad's lab in 2012 for my PhD, incidentally, the foxtail millet's genome was released by two independent groups. One was from JGA and the other one was from BGA. So what we did is, like, we took up the data and we started looking into, what actually foxtail millet genome has. One interesting factor which we noticed in this Chinese paper, this BGI one, is that they showed that around 1,500 genes are unique to foxtail millet. They say that these genes are not present in any of the other crops. And out of these 1,500 genes, they have annotated 586 genes as response to water. So that was the catch. We started looking into, or we started characterizing these genes. So when we re-annotated ourselves manually, these 586 genes, we found that these genes encodes for different, gene families belonging to transcription factors, stress response factors, etc., etc. I'll show you one example of this HSP because two people were involved in this. That time, A girl from Maulana Azad National Institute of Technology, Bhopal. Her name is Janani. She joined for her MSc MTech internship, and Roshan Kumar Singh, he is from University of Calcutta. He joined for his PhD. So we three were there, and we started working on characterizing each and every gene families. And one example I will show you, like this HSP1, because that work is being carried forward, and even a patent has been filed. So although I will not be able to give much details, because Dr. Manoj Prasad will be giving more details on this, about the progress that is being done, but I'll show what we have done as a preliminary, or as a base for this work. So we all know that HSPs regulate different aspects of stress defense through protein-protein interactions of the target proteins, of course. And what we did, we went to identify the genes that encode for these HSPs. So we all know that HSPs fall into 5 different categories, but the data of HSPs present in other related species were available, but in foxtail millet there was no report. As I said, we were the first to do that. So first thing what we did is, like, we tried to identify the heat shock protein encoding genes in foxtail millet using several computational tools. We identified 100 plus genes. We classified them according to their molecular weight, and we analyzed this phylogeny of the heat shock proteins, which classified the proteins into different classes and subgroups. I'm not going into those details. And when we plotted them on the 9 chromosomes of foxtail millet, we found that these genes are ununiformly distributed on the 9 chromosomes, and few were segmentally duplicated. The colored lines indicates the segmentally duplicated genes, and a few were tandemly duplicated. The yellow ones indicate the tandemly duplicated genes. So by the time we had an RNA-seq data of 4 tissues, namely root, leaf, spica, and stem, and we have the library of foxtail millet transcriptome. So we checked the expression of all the 100 plus genes in this library, and which showed a differential expression pattern, as you could see. And from this, we chose 34 or 37 candidate genes for further study. So immediately what we did is we chose 2 cultivars. These are contrasting cultivars, IC4 and 41. IC4 is tolerant to stress, whereas IC41 is susceptible. So we checked the expression of these 37 genes in 2 contrasting cultivars subjected to different stresses, namely heat, cold, salinity, and dehydration, and hormone treatments like abscisic acid, methyl jasmonate, and salicylic acid. Although you could not predict anything from this heat map, one thing I can show you in specific is that this one gene, SHSP27, showed 35-fold higher expression in the tolerant cultivar during heat, and 47-fold higher expression in the tolerant cultivar during salt stress, and 21-fold higher expression in cold stress in the tolerant cultivar. So the gene is the same, but it is differential expressing between 2 contrasting cultivars, where it is showing several fold upregulation during heat, cold, and salinity in the tolerant cultivar. So we did 2 things before publishing this out. One thing is that we tried overexpressing. By the time foxtail millet transformation system was not available, one of our RAs, Dr. Priyanka Sood, her paper was published last January, but till then we didn't have a stable transformation system in foxtail millet, so we had to do all our experiments preliminarily in yeast and then move to rice. So in yeast we tried overexpressing this SHSP27 gene, and you could see that this transgenic cells overexpressing SHSP27 showed enhanced tolerance to heat, salt, and dehydration stresses. And now, although we published it with this data, then Roshan took it up for his PhD, and what he did is, like, he tried overexpressing this gene in the rice. You could see, although the data is very preliminary, because this part will be covered in detail by Dr. Manoj Prasad in his next part of the talk. You could see that transgenic lines are showing tolerance as compared to this wild type when the irrigation was withdrawn. So in addition to that, he also observed that the transgenic lines showed improved agronomic traits, including height, tiller number, panicle number, size, number of seeds per panicle, etc., etc. So by this way, what we did is like when we established our, I'm not going into these details, we established an integrated pipeline for gene family analysis, and we identified several such uh, candidate genes from that 584 genes which were annotated as response to water. And we are now raising, or now we are functionally characterizing each of these genes. And very recently, like I mean, it is in May, we also studied the C4 photosynthetic genes in foxtail millet and its wild relative Setaria viridis, and we have published that in this Journal of Biotechnology. So now what it did is our analysis has opened an avenue for further research in foxtail millet for characterizing each of the candidate genes. There are several reports now available where people are showing that our genes are responsible for they promote tolerance to salt. In one nice study published in Frontiers in Plant Science, they have shown that a gene promotes, or a gene confers drought recovery even after withholding the water, even after withholding this irrigation for 20 days. So such things were like, I mean, there in the foxtail millet genome, and we try to unlock that potential. And then we realized that many of these genes which were annotated as response to water by Zhang et al. Were transcription factors. So we thought like, I mean, why shouldn't we identify all the transcription factors encoded in foxtail millet genome? So we developed an integrated pipeline. Then we identified 2000 plus genes belonging to 55 different transcription factor families. And then we didn't stop there. We kept that in a foxtail millet transcription factor database, and we kept it open for the global research community to access and use the data further. On the other hand, one of my seniors, she also studied the microRNA part. Now we have moved very advanced. Like now we are studying all the small RNAs, all the long non-coding RNAs. We are even studying how each of the genes respond to particular stress. But these are like, I mean, very preliminary things which we did, which stands as a base for any further studies by us and by the global research community. And in one such study, we were the first to report dehydration responsive mRNAs in foxtail millet, and that was published in Planta. Then we have also constructed a foxtail millet microRNA database, and we put all the data in the database. So by this way, whatever we did, or whatever I did during my PhD, or whatever my seniors did during their tenure, we never kept anything to ourselves. We put it on an open access database, and we kept it open for the researchers to access and use these resources. And by this way, it has helped several researchers to work on genes, work on microRNAs, work on markers which we have identified. And we are now getting several papers coming out from different laboratories around the globe, studying different aspects of foxtail millet. Even very interesting studies are being published in the line of using foxtail millet as a feed. Also, as in the feed, they are trying to use foxtail millet as a replacement to oats, where they have shown that few of our genes which we have identified before are playing role in enhancing the biomass or the digestibility is also. So this is the thing. Like, I mean, in 2013 when we started, there were only few markers and few genes were characterized, and the crop was remaining neglected in terms of research. And with the year, what we did, we added several markers, and we characterized several genes and gene families. And whatever we did, we put it in open access databases, and we kept it open for the global research community. By this way, we transformed a neglected and underutilized crop into a crop with rich genetic and genomic resources. And we didn't stop then also. Like, I mean, several of our students, both at NIPGR and University of Hyderabad, we are working on each of these genes, and we are also using these markers to look into different traits. Like say, for example, in millets, there is a big issue of rancidity. Millet flours won't stay on shelf for more than 7 days because it goes rapidly rancid. So we are also working on that aspect. And millet has appropriate amount of anti-nutrients, like for example, phytic acid. So we are also working in such aspects to develop low phytic acid lines. And we are also looking into introgress, better qualities or better traits of foxtail millet into other cultivated millets as well as rice through both breeding and transgene based approaches. That is how the things are progressing. But the stone was laid by us, onto which a huge building is being constructed. And as you could see, with the year we made this progress, tremendous progress. In the year 2014 alone, we published 13 papers on foxtail millet from a single lab, and that is how we worked on this crop. And I think I can stop here by showing you, like, I mean, by after that we also wrote an article where we showed, like, I mean, how this information can be used to develop or to improve or to develop such genetic and genomic resources in other neglected crop species. And we were also the first to organize a national conference on neglected and underutilized crop species for food, nutrition, energy, and environment last year, August 2nd, at National Institute of Plant Genome Research. Dr. Manoj Prasad was the convener and I was the co-convener, where we brought all the research community who are working on such underutilized crop species to NIPGR, and we had several brainstorming sessions, talks, poster presentations, and we tried working on this further. We had Dr. Renu Swarup from DBT and Dr. Mohapatra from ICAR. They both also assured that the funding agencies will also work out on how to promote these underutilized and neglected crop species, because these species hold the potential for addressing this hunger and malnutrition. So in that direction, we are working forward. And with this, I complete my talk and I can address the questions, if any.
Thank you. Thank you for your talk. As you say, is that it's a first part, and we are expecting Monu sir giving the more details and more about your lab work.
Exactly.
And I am request to you to if you because we are waiting for the answers and it's coming through. There is a box, it's Q&A. If you see it, then there is a 2-3 questions. Can you go for it? And I am collecting few answers from the YouTube if it is suitable and exactly for your talk. Have you got the Q&A box?
Yeah, Q&A box is there.
Yes.
Okay, definition of markers. That is previously it was like a 2-mark question, but now it has become broad. Once we were saying that these won't code for anything. Now when SNPs or functional markers came into picture, the definition changed. There is a dynamic definition for markers which changes from time to time. As of now, how we say is like, I mean, this is a genomic region which regulates a particular trait. So that is what, like, broadly we can define a marker. Say, for example, these SNPs, right? It can do wonders. So those are, now we have moved to SNPs. Previously, SNPs was not in the picture because we were not able to sequence the genome. That was limitation in funding, etc., etc. So this definition changes with respect to even markers. If we are using SSR, the definition is different. If you are using SNPs, the definition is different. DD-RAD sequencing. Yeah, usually what we do is maybe if you should be having a little knowledge on Illumina sequencing to understand this DD-RAD. In Illumina, what we do, we isolate the DNA, we fragment it or we shatter it, then we ligate the adapters and then we go for sequencing. In DDRAD, what we do is like, I mean, we use restriction enzymes. One is a frequent cutter and one is a rare cutter. So frequent cutter and rare cutter, as you know, restriction enzymes cuts at a specific part, I mean, at a defined sequence, like TAACC, something like that. So if it cuts there, it will be easy for us to ligate the adapter, and then we can go for sequencing. So by this way, we were able to enhance the coverage using this TD-RAD sequencing. It is the definition itself is like, I mean, double digest restriction associated DNA sequencing. Double digest means we have to use two different restriction enzymes to digest the DNA and then go for regular sequencing. We have foxtail millet germplasm of different colors. Does color has some specific advantages in terms of quality, like raggy? Yeah, like, I mean, one of our MSc student is working on this. She is trying to see whether the color has something to do with the content of the millet, and now we don't have much data on it. Once it comes, maybe we can discuss on this actually. How secondary cell wall related with stress tolerance? Secondary cell wall we studied because that time, I mean, foxtail millet is closely related to this biofuel crops. So few groups which were working on this biofuel traits, they said that can we look into some genes which has role in secondary cell wall, lignin biosynthesis, etc., so that they can use that information in their crop. Because two groups, one is working on switchgrass and the other one on napier grass, it was their request. Then we went on to look into the genes encoding for this secondary cell wall biosynthesis genes, and that is how that paper came up. Linkage drag will change the efficiency of marker-assisted selection. We didn't go up to that point where, I mean, we never went to the field, because to be frank, in NIBJ there was no experimental field where we were able to do this kind of field level experiments. So we didn't go up to the level of marker-assisted selection. Once we go there, then only we will be able to understand how much this linkage drag has impact on the efficiency of mass. Why finger millet is not used common for cooking as rice? This is a very interesting question. Like, I mean, people nowadays are asking like why we are not using millet in the place of rice, because several millets are very bad in taste, to be frank. You have to put little more effort to cook it properly, or cook it in a tasty way. Other one is rice has that the taste of rice cannot be replaced, right? Although barnyard millet, you people would have heard of. There is one restaurant in Gurgaon, I forgot the name. There you will get barnyard millet biryani. So now people are like, I mean, coming up, people are moving towards millets. But as I said, one thing is popularity, it is lacking, because only a marginal population is consuming this millets. Even people, if I see, I developed my own interest on why I wanted to see what is there in the literature. So we had a tie-up with one PhD student who was from Department of Tamil, and that fellow went and searched the Tamil literatures, and he came up with a statement that millet was there before rice in Tamil civilizations, because in many of the songs which were written that time, he is founding the inscriptions of millets than rice. So it was there once on our plates, and slowly we moved to rice maybe. And now few people are coming back to millets, but mostly what is happening is like we have deviated towards oats. If people prefer oats more than millets. So yeah, there is also an issue. But slowly the awareness is coming up. Slowly the people are developing protocols. Even recipes are coming up. So we have to wait and see how much millets can gain on the plates, gain space on the plates. ILPs are intron length polymorphic markers. It was once very famous because before this SNVs came into the picture, because in the introns there are polymorphic regions, and we developed primers flanking those introns, and we were using those That was very much useful in several of our genetic diversity studies, but now it is more of outdated one. Resistance genes from foxtail millet can be transferred to rice. We have to see whether what we can do in this regard. It's a long way to go. Good markers for finger millet. Now finger millet genome is available, so you can easily jump in and identify genomoid markers. If the genome sequence is available in SRA database, take it out and put it in MISA, you will get thousands of SSRs. If 2 cultivars were sequenced, I believe that different cultivars of finger millet has been sequenced at least in transcriptome levels. If that is so, you can align the reads of different cultivars to identify SNPs, and of course these SNPs will be gene-based SNPs. So these SNPs have more value than the genomic SNPs. So you can go for it. Only thing is, you have to have a little bit of knowledge in this NGS thing. Will your team be doing research to identify genes of foxtail millet that is responsible for its yield? Yeah, we have identified several genes and we are working on it. Since the data is in very preliminary stage, I have not shown it. We are on it. Production and productivity of small millets is low. How can we promote to cultivate these crops? Production and productivity is low because it has not been in the mainstream research. If research would have been done, then production would have, or productivity would have been enhanced. Say, for example, if you get any chance to travel to Madurai, the southern part of Tamil Nadu, there is an agriculture center there. If you go there, they have released a variety of barnyard millet called MDU. You won't believe me, people are farmers who were cultivating rice in that Madurai region are now shifting to barnyard millet because that is giving them more reward. So very few people or very few group are actually working on this yield or productivity parameters, and that is why we are not having good varieties which has more yield. So before promoting, maybe we should work little more on enhancing its traits. Is it possible to cross foxtail with finger millet? Foxtail millet with finger millet, I don't know whether it is possible to cross because we have not tried that, number one. And finger millet itself is, pearl millet itself is a hardy crop. I confused with the before question actually. For crossing foxtail with pearl millet. Pearl millet itself is a hardy crop, so transferring any gene from foxtail millet to pearl, I don't think it makes relevance because I think both are equally good in terms of performance and climate resilient traits actually. What are the profitable millets for farmers in UP? You have to see, in that case, what we try to advise people is you have to go to the nearest agricultural university where they will be having such data and they can clearly guide you and they can even give you seeds which you can use for cultivation. So it should be dependent on your climatic conditions, soil, rainfall, etc., etc. So such institutes or universities can help you in this regard. There is CRISPR tool rather than molecular markers to create gene targeted crops. What about your stand? The purpose is totally different. Markers is to identify genes. CRISPR is to manipulate the genes. So can we create CRISPR biofortified millets to silent transidated genes? Yes, we are using that approach. We will see how much success we got sooner or later. What is C4 panicard model for analyzing stress responsive gene? So far, among the C4 panicards, foxtail millet's genome is out, pearl millet genome is out, finger millet genome is out, sorghum genome is out, maize genome is out. These 5 crops are to an extent climate resilient or stress tolerant. So you can study the genomes of all these 5 crops to identify what these crops have gained and what the other crops have lost. Certain studies use promoter sequences of foxtail millet for transgenic studies. especially for organ-specific expression. What makes the promoter sequence of foxtail millets important? I have not I have not come across any such studies where they have actually used promoter sequences of foxtail millet in transgenics. So if I could see that, then I can answer. Otherwise, I don't know. Yeah, that's all we have. Your mic is muted. Yeah.
Yes. Thank you. Thank you for the this answer. And from the question, you can understand that there is a variety of people, and
Yeah.
some of the known to your field and some of the new to your field. And we assure the audience that as our speaker says that it is a orphan crops, and nowadays the orphan crops has very good futures, because the environmental conditions is changed, and these crops are very tough, and they can adjust very suitable in these hard conditions. Rajiv Bhasne had also very frequently used these terms for the chickpea, raisin pea, that these are the orphan crops. But as I know that the orphan crops give the huge field to the research, and it's a very open field. If you work for our orphan cause, your paper will be good, your citation will be good, and ultimately you contribute more in the society. And tomorrow we also have a talk with Tara Satyavati ma'am on the orphan
Yeah.
cause. Also, we have a talk on the your side that on this platform. So I think after few of your talks and others talks, palm millet is going to be very known, and due to availability of genome sequencing is very easy to nowadays to work on that, like CRISPRs and others things which are booming nowadays. So I'm very much sure your future work, your gene from the palm millets to the rice or any other cereals will help them to adjust in the field. They help to ensure the food security ultimately, because we have to ensure that how 10 billion people going to feed it by in near future. So I just end this talk here, and we go for the a short interview to you. It's for our audience, mainly those are the MSc, JRF, and early career. So you free, you don't feel there is some special to you, and your career is so fascinating to our also. Is a very short time you gain very different platform. I hand it over to Shoma to doing a short interview to you. Shoma.
Hello, sir.
Hello, ma'am.
It was a beautiful, very nice talk, sir. So I'm starting the interview session now. Sir, my first question to you is that you are quite a successful researcher, having achieved so much at a relatively young age. What was your experience like? What things did you face when you built up your career? Could you please tell it to your young viewers?
What made the difference in my career is NABGR.
Okay.
So I did my BSc and MSc from Madurai Kamaraj. It was an affiliated college to Madurai Kamaraj University.
Mm-hmm.
And during my MSc final year for this MSc dissertation program, I came to NABGR to join Dr. Manoj Prasad's lab. To be frank, that was the first time I'm hearing that there is an exam called CSIR net, which has to be qualified for getting into PhD in such institute at the final year of my MSc. So that was the kind of awareness we had in the southern district of Tamil Nadu. Then it was Dr. Charulata who explained to me, like, what are the exams that has to be qualified for doing PhD in such a reputed institute, so and so forth. Then I went back to Tamil Nadu. Then I attempted once, and then I thought, like, no, it's not going to work for me, so I'll better go to a different field. But then I received a call from Dr. Manoj Prasad. Sometime he just called to to know what I'm doing right now. Then he has told like, I mean, why are you giving up? I can take you for PhD in my own lab, since you are already a trainee, you know the lab well, you know the institute well. You just qualify that exam and come. And it was his word that made the difference. I qualified CSNet twice, and then I told him that, sir, now I have qualified. Can I come? Then he said, like, immediately come. Then
That's
I went
great.
there for PhD. One thing I realized in my journey is that it's not just the degree that is going to make a difference in your career. It is also the place from where you get the degree. If I would have
So
done my
true.
PhD at MKU, I wouldn't have reached this point to this point.
So true.
So when I went to NAPGR, it of course gave me that exposure, right? I got several publications. I got Fulbright. I got Indo-Australian Career Boosting Gold Fellowship. I got INSA. I got NASI. I got etc. And everything is hanging up behind me. But
Yes, sir.
this kind of thing I wouldn't have got if I continued maybe in a state university. So yeah, that is what even I tell people, that uh, go for a national institute where funding is ample. Of course, funding is ample, right? They have, of course, they get a good amount of funds. They have a very good infrastructure. And whatever the fellowship or whatever an opportunity comes, the first information comes to such institutes. So those students will get the information on such programs where they apply. And with time, these institutes have created a label. So yeah, that helps people in climbing up their career ladder. So that worked very well for me. And of course, it was purely because of the support of Dr. Manoj Prasad, who will be soon talking to you people on this platform. So
Yes,
you can
sir.
ask the same question to him too.
Okay. Okay, sir. My next question is that in the era of big data and artificial intelligence, how do we progress with um respective to plant research and its field applications?
Yeah, in research, this big data and artificial intelligence are already there. But in field applications, I think they have not been used to the fullest. For example, we still do not have an integrated platform like an online device or an app to inform farmers about disease attack or weather conditions or soil health, etc. Another aspect which has tremendous scope is the predictive agricultural analytics, where artificial intelligence could be used to predict the optimal time for sowing, updating the farmers about irrigation parameters, application of fertilizers, and apps should also predict the optimal harvest time, because if it is going to rain tomorrow, then the farmer should harvest today itself. So those things, we have seen many people or many farmers who go into loss because the final day of his harvest, it will rain and all the crop will fall. So such things will be highly useful in field where progress has to be made actually.
Yes, definitely. Sir, my next question is that according to you, which sectors of plant science research and agriculture will prosper in the near future?
Let me split it up into two, in agriculture and in research. In research, we are now using this integrated omics approaches. Previously, we were focusing only on individual omics tools. If you could see, my talk was purely on genomics, but now we are realizing that, okay, working on or focusing on one particular aspect is not going to help, or it's not going to give the complete insight of what we are trying to address. So we have to integrate different approaches. So integrated omics is now coming up, and we already have newer technologies like speed breeding. I'm developing one chamber in my greenhouse, which actually has revolutionized this crop improvement programs. And now even seed companies are using speed breeding approach to produce quality seeds in shorter duration. Papers are coming up on coupling this transgene technology with speed breeding and speed breeding. And these are few prospective areas which I believe that would come up well in the future to address this food and nutritional insecurities. And in agriculture, if you ask, we again and again talk about this mainstreaming underutilized crops. If you could see, rice, wheat, and rice, wheat, and maize. These three these three crops serve 50% of the world population, where our world population is 7.7 billion. So there is a huge inequality between the available crops and the population or numbers to be fed. So that is where we always talk about this and mainstreaming this underutilized crops, because many of crops are edible. Our archaeological evidences shows that approximately 7,000 crops were cultivated by our ancestors, which now got narrowed down into 3 plus another 12, which along with 5 animal species, which is catering 90% of the world population. So now people are slowly getting awareness, and this mainstreaming underutilized crops or research in underutilized crops is slowly gaining this momentum. And we have also started realizing that these species might be not just millets. There are several legumes that are underutilized, tubers, leafy vegetables, etc., etc. These have the potential to tackle this hunger and malnutrition, and that is what is coming up in these days.
Okay, sir. Sir, my final last question is that what would you advise to the young research fellows out there? How they could make their careers better, or how they could cope up with the pressures of research?
Yeah, first and foremost thing which I tell to a researcher is that they should have awareness. Awareness in professional front and awareness in personal front. In professional front, they should be aware of what is going on in the current field or in their field of research, what is going to come up next, and in what speed we are working compared to what is being done by our competitive labs or collaborative labs. That comparison will keep us motivated. That comparison will make us run, because when I entered for PhD, the first thing Manoj told me was PhD is not a journey, it's a race. If you have to run fast to achieve, to get the degree fast. So that is where we started running, and we ended up here, right? So that fetched us papers, let it be research paper, review articles, book chapters, popular articles. A student should not hesitate to write. Here, writing and research should go, hand to hand. So that one thing everybody should realize. Coping up with pressure is like, I don't know what kind of pressure people get, but in our lab, maybe particularly me, I never felt any pressure because I do my job. That's it, right? PhD is an objective-based research. It's not a timing-based research. Your supervisor will give you 3 objectives which you have to complete for preparing your dissertation. So if you finish up your objectives fast, you will submit fast. If you don't finish, you will be late. So that is what is happening. Yeah, pressure will be there in terms of publication. Pressure will be there in terms of this writing and these things. So one has to be, competent enough to tackle those things. On personal front, the awareness is like, there are lots of fundings and fellowship opportunities available. May it be national level, may it be in international level, even state level, several state governments like this Kerala state, they have a Kerala Science and Technology Council or something like that. They give some, benefits or fellowships or funding opportunities for researchers, early career researchers and all. So that awareness is very much important, because those things should be added in your CV. That makes your CV strong. Not just completing a PhD with 1 or 2 papers is going to help you. So that is what, to make a difference in the CV, they should work, and they should have this complete awareness. Awareness is the first and foremost thing a student should have in this regard.
Yes, sir. So thank you, sir. I'm sure many of our viewers, the young viewers who are pursuing PhD, they got many useful tips and advice from you. Thank you, sir. It has been a wonderful opportunity for us to have you here. Thank you so much. I hope we will see you again in the future soon.
Thank you. Definitely.
I'm giving it over to Shubho.
Thank you. And I also include one thing: Fox millet genome sequencing also help you a lot.
Exactly.
Because in your time during the PhD, if it is this,
it came up.
Yes. Another is speed breeding. Actually, I'm trying to communicate Lee Hickey from Australia for the these purposes.
Mhm.
Let's see if it is possible to hear from him. So it's very nice to all of us, because now speed breeding is an opportunity to us to rapidly generate our
Yeah.
crops. I'm working
They.
in I'm working on jute, so I know that it
Okay.
takes 1 year.
Yeah.
So
So you need a chamber.
I don't know what is it possible for this type of, because I am 6 feet and jute is about 14 feet, or average is at 12 and 13 feet.
Okay.
So it's very difficult to in chamber, but let's see what happens.
Yeah.
And we are very anxious to hear you again in the future, because now the time where the genes are incorporated into the crops, and the crops can be engineered in different way. So we are looking forward to your future work, and we expect you once again in future. Thank you.
Definitely.
Now we just giving some instruction to our viewers to how to collect and how to submit their application for the certificate. So you may lead this meeting, and I
Okay.
give
Okay.
you
Yeah.
best wishes for your
Thank you.
work.
Thank you. Thank you, everyone. Thank you.
Okay, thank you. Uh, so now I'm request Soma to give some instruction to our viewers, and I assure you that the last webinar we have distributed certificate within a day, in 24 hours. So we changed our system, then it helped us, but two previous two webinars, we mailed every certificate to individual person. That's why it's taking so long. But within 2 days, I think within 2 days, we are going to finish these things. And similarly, we have to arrange our future talks. So we are also busy with doing that. That's why it takes time. But like last webinars, our certificate also available in our website. This For this webinar, we uploaded in our website. We got various mail that your certificate is not there, but I'm sure we just go through the applicant list, check them, printed their name on the certificate, count them, and ultimately upload the all file. If it is not found, if you not found it, it is problem of your internet connection and your Google account. So so please not send us such type of mail that my certificate is missing. If your name in that list, your certificate in the drive, there is no ambiguity in that section. Check your internet connection, sign into the Google. It may require sign into the Google Drive also, and collect it. And we personally not send one certificate for the third and onwards webinar, because we are busy to submit first and second webinar certificate. We build a system to all of you, go to the shared drive and found your certificate. If there is any problem, check first yourself. Is it problem on your internet? Is it problem of your drive or your Gmail account? If you can't solve it, go for a friend who knows these systems. But don't mail us or communicate us for this reason. We need a system, because we throughout the year we're going to present and the webinar. So if we give our time to these things, it hamper our upcoming webinars. So tomorrow also we have a webinars on millet, but it's not the foxtail millet. It's a pearl millet. And I think ma'am will be discuss about the biofortification, food habits, new foods, etc. So let's join us again in tomorrow's talk. I hand it over to Shoma to give few, I think all of you know, but few instruction here.
Hello. Okay, I'll share my screen so that they can see how to download the certificate. Is it shared? Okay. So on my screen, you can see the website, right? So after every webinar, wait for 2 days and then go to the website. Scroll down to the bottom of the page. Can you see this bioingene.com live webinar certificate application list? I hope you can. Here, there are 4 lines. Okay, the first one consists of bioingene.com live webinar certificate application procedure. This is the procedure that explains how you will apply for the certificate. By now, I think many of you know how to apply for the certificate. After you're applied for the certificate, we will get the list. That list is uploaded here. That these are the names of the applicants. This is the list. See, the first one is on the first two lists. The first two are different. The first two are for CRISPR and chickpea. That is, we are mailing these certificates to you. But from the third one, that is from the 6th July webinar onwards, we have devised this new system. Here, you will get your own certificate. Get your own certificate from this website. You don't have to wait for us. Click on the link. Now, can you see there's a Excel sheet? On the first column, you can find the page numbers. This is your file number and your name. Refer your name, find your name, and because it is presented here alphabetically, you won't have a problem to find. It is all in alphabetical order. Find out your name and then what is your file number. If you get your file number, then go back to the website and click on certificates. 6 July 2020, development of climate change resilient rice varieties through molecular breeding by Raman Meenakshi Sundaram. And then you can see the link for certificates for this particular webinar. Click on certificates. You will get into the drive. Here you can see I'm already inside the drive. In the folder, 6 July webinar, webinar number 3 on 6 July. You can see all the certificates are here and they are named as page number. Okay. Find your page number and download your certificate. Yeah, one issue can be that you are doing it from your mobile phone. There could be an issue because maybe you don't have a good speed for your internet. You need, because it is a drive and so many pages are there. It will take time to load, and maybe if it is not loading, you are thinking that okay, these are pages are missing, but no, give it time, it will load, or get a good speed, high speed internet connection, so that you can download this. Like you can do it from your mobiles or you can do it from your laptops, but wait for some time so that the page loads. The page will load, the drive will have all the certificates. Find your see there are how many certificates? Many certificates. See the page is loading slowly and scrolling down. It is keep loading. So give some time. It will load. You download your certificate. So that's so convenient. Okay. I hope you all got it. And regarding tomorrow's webinar, I'm really excited for tomorrow's webinar. We will have our first woman speaker for this session. She is Dr. Tara Satyavati. She'll be speaking on her journey from biofortification in food crops to including nutritional traits in varietal identification and release in pearl millet. See you all for this much awaited webinar. See you at 11. Registrations are open, but I don't think they'll be open for long. Please hurry. If you are still not registered, please hurry, because otherwise you won't be able to apply for the certificates. Please hurry for the registration. And also keep an eye on the notice board here on our website homepage. At the top, you'll find there's a notice board. We keep putting in information from time to time. We keep updating the notice board. Please have a look. You'll find that sometimes we have put in new information. Please come and see, come and check. Shubha wanted me to give instructions in Hindi and Bengali. The only two other languages that I know. I don't know why. I hope all of you know English, but still, if you want, okay, it will be fun. Let's see if I can do it right. Students, okay. Hello. This is the instruction part of our webinar. I'm trying to speak in Hindi and it's not coming. नमस्ते, आप सबको स्वागत है इंस्ट्रक्शंस में वेबिनार के। हमारे वेबसाइट पर जाइए। हमारे वेबसाइट के नोटिस बोर्ड पे आपको नोटिस मिलेंगे। यहाँ पे आप हमारे जो इंफॉर्मेशन रहते हैं सर्टिफिकेट के लिए वो यहाँ पे पोस्ट किए जाते हैं। एंड सर्टिफिकेट्स के लिए आपको वेबसाइट के अंत में जाना पड़ेगा, स्क्रॉल करके नीचे यहाँ पे एक हेडिंग है bioingene.com लाइव वेबिनार सर्टिफिकेट एप्लीकेशन लिस्ट। यहाँ पे आपको लिंक मिलेगा सर्टिफिकेट के एप्लीकेशन के लिए। जो प्रोसीजर है, जो तरीका है सर्टिफिकेट के अप्लाई करने के लिए, वो पहले लिंक में दिया है। और इसके नीचे जो 6 जुलाई का जो वेबिनार था उसका जो सर्टिफिकेट है वो मिल रहा है आपको, आप खुद ले सकते हैं। आप इस लिंक पे क्लिक कीजिए, आपको एक लिस्ट मिलेगा जो कि उस लिस्ट में सारे एप्लीकेंट्स के नाम हैं। उस नाम के पास हर नाम के लिए एक पेज नंबर है जो कि फाइल नंबर है, एक पीडीएफ फाइल है उसका नंबर है, उसका नेम है वो कि पेज नंबर है। जैसे कि पेज नंबर 1 से शुरू हुआ है। यहाँ बहुत सारे सर्टिफिकेट्स हैं। आप इसमें अपना नाम ढूंढ के जो पेज नंबर है वो ढूंढ लीजिए और फिर से वापस वेबसाइट में जाइए। वेबसाइट में आपको एक और लिंक, उसी लाइन में एक और लिंक है, सर्टिफिकेट्स लिखा हुआ है, उस पर क्लिक कीजिए तो आप सीधा ड्राइव में चले जाएंगे। हमारे जो गूगल ड्राइव हमने शेयर किया हुआ है वो गूगल ड्राइव आपको दिखेगा। थोड़ा टाइम दीजिए इसको लोड होने में और अपना जो नंबर आपका मिला है लिस्ट से, आपके नाम के पास जो नंबर था पेज नंबर, वो ढूंढ कर निकालिए, उसको डाउनलोड कर लीजिए, वही आपका सर्टिफिकेट है। इस पेज को लोड होने में थोड़ा वक्त दीजिए। आपको आपका सर्टिफिकेट, अगर आपके लिस्ट में आपका नाम है तो आपको डेफिनेटली आपका सर्टिफिकेट मिल जाएगा। हमारा अगला जो वेबिनार है वो डॉक्टर सीतारा सत्यवती मैम का है। आप जरूर कल आइए, वेबिनार देखिए। हमको बहुत दिनों से इंतजार था इस वेबिनार का। कल सुबह 11 बजे फिर से मुलाकात होगी वेबिनार के जरिए। थैंक यू। Shall I continue in Bengali? Okay, for my Bangladeshi friends and people of West Bengal, definitely, and all over the world, wherever you are, all the Bengalis out there. নমস্কার, আমাদের ওয়েবিনার আশা করছি আমাদের ওয়েবিনার আপনাদের ভালো লাগছে। আজকের ওয়েবিনারটা ছিল ডক্টর মেহনাথান মুথুমালিরাসানের স্যারের। উনি আজকে বললেন ডেভেলপমেন্ট অফ জেনেটিক অ্যান্ড জিনোমিক রিসোর্সেস ইন ফক্সটেল মিলেট, প্রোগ্রেস অ্যান্ড প্রসপেক্টস নিয়ে। এই ওয়েবিনারটা যদি আপনি অ্যাটেন্ড করেছেন তার সার্টিফিকেটটাও আপনি পেয়ে যাবেন আমাদের ওয়েবসাইট থেকে। এর আগের ওয়েবিনার নাম্বার 3 যদি আপনি অ্যাটেন্ড করে থাকেন এবং তার জন্য অ্যাপ্লাই করে থাকেন, তার সার্টিফিকেটটা আপনি আমাদের ওয়েবসাইট থেকে পেয়ে যাবেন। আমাদের ওয়েবসাইটের একদম নিচে স্ক্রল করে গিয়ে দেখবেন bioingene.com লাইভ ওয়েবিনার সার্টিফিকেট অ্যাপ্লিকেশন লিস্ট দেওয়া আছে। সেখানে প্রথম লিঙ্কটা আছে কিভাবে অ্যাপ্লাই করতে হয়, সেই প্রসিডিওরটা বলা আছে। আর 6 জুলাইয়ের যে ওয়েবিনারটা হয়েছিল সেটার লিঙ্কটা দেওয়া আছে। এই লিঙ্কটাতে ক্লিক করলে আপনি একটা এক্সেল শিট পাবেন। সেই এক্সেল শিটে যদি আপনি অ্যাপ্লাই করে থাকেন ঠিকঠাক, আপনার নামটা এখানে আসবে। যেহেতু অ্যালফাবেটিক্যাল অর্ডারে এটা দেওয়া আছে, আপনি আপনার নিজের নামটা খুঁজে বার করুন। তারপরে আপনি পেজ নাম্বারটা দেখুন, এর পাশে একটা পেজ নাম্বার দেওয়া আছে। সেই পেজ নাম্বারটা হচ্ছে আপনার ফাইল নাম্বার। এই নাম্বারটা আপনি খুঁজবেন আমার ওয়েবসাইটে। আবার এই একই যে ওয়েবিনারটার ব্যাপারে বলা আছে 6 জুলাইয়ে, সেই লাইনে দেখবেন সার্টিফিকেট বলে একটা হাইপার লিঙ্ক আছে, এটাতে ক্লিক করুন। এইটাতে ক্লিক করলে ডাইরেক্ট আপনি ড্রাইভে চলে যাবেন, আমাদের যেটা আমরা শেয়ার করেছি গুগল ড্রাইভটা। সেই গুগল ড্রাইভে একটু সময় দেবেন পেজটা লোড হতে। অনেকগুলো সার্টিফিকেট আছে, একটু সময় তো লাগবে। এবার আপনি আপনার যে নাম্বারটা আপনি পেয়েছেন সেই এক্সেল শিট থেকে সেই নাম্বারটা খুঁজে বার করুন, পেজ নাম্বার। সেই নাম্বারটাই হচ্ছে আপনার সার্টিফিকেটটা। ডাউনলোড করে নিন। আর একটু অপেক্ষা করতে হবে না আমাদের জন্য, কবে আমরা মেলে পাঠাবো সেসব অপেক্ষা আর করতে হবে না। ওয়েবিনারটা যে দিন ওয়েবিনার হবে তার ঠিক 2 দিনের মাথায় আপনি পেয়ে যাবেন আপনার সার্টিফিকেটের লিঙ্কটা। 2 দিন আমাদের সময় লাগছে, আমরা নিয়ে রাখছি হাতে সময়, যাতে আমরা এই লিস্টটা তৈরি করে ফেলতে পারি, অ্যাপ্লিকেশনের লিস্টটা তৈরি করে ফেলতে পারি। এই লিস্টটা আমরা তৈরি করে ফেললাম, সার্টিফিকেট গুলো তৈরি করে ফেললাম 2 দিনের মধ্যে। তারপরে আমরা ওয়েবসাইটে এটা আপলোড করে দিচ্ছি। থ্যাংক ইউ।
Hello. Hello. Officially, this webinar is the end. We give the instruction to how to collect the certificate. See you tomorrow at same time. I'm requesting all of you, those are the new on today's YouTube channel, please subscribe this channel, and if you subscribe, then it automatically gives you the notification when we are in live, because our all webinar are live hosted in this channel particularly. Very soon we're going to upload today's webinar talk and the interview in the different separate file in this channel. We also going to upload yesterday's webinar talk in this channel. And I'm expecting all of you once again for tomorrow's webinar on millet by Dr. Setaria ma'am. So I'm just end this webinar. Thanks to all of you. Thanks to our volunteers. Those are very helpful to us to distribute the certificate, to help to tackle the chat box during the webinar. They answered a few of your questions. I'm also thankful to our team, Bioingene team, thankful to Neha, thankful to Shoma, and thanks to our speaker to choose this platform to share his research. Now I'm going to end this session. See you tomorrow.